Dear friends and colleagues, it's a great pleasure to open another episode of the ESDR Kitchen Freshly Baked, which is what you know, the research highlights, recent research highlights presented by young colleagues. And today we'll have a focus on professional and semi-professional antigen presenting cells. We'll directly be starting with the first presenter, Raquel Sal-Carro, who did her master in biology in Barcelona, and since 2021, she's researching autophagy and macrophages, tissue-resident macrophages in Strasbourg. Please, Raquel, we don't want to lose any time. Give us your highlights in research.
Hello, everyone. Thank you for your presentation. So I'm very happy to be here to be able to talk to you about the paper that we recently published on the role of autophagy for the maintenance of epidermal Langerhans cells. So autophagy is a very important process for the cell because it allows it to degrade components of the cytoplasm and then recycle the basic building components. So this is going to be a mechanism that is activated under situations of stress, such as nutrient deprivation, and allow the cells to adapt. We know that autophagy is very important for the regulation of the immune system. In particular, for antigen-presenting cells, we know that autophagy regulates cytokine production, antigen presentation, and it can also regulate the inflammation response. And we also know that this is, although it's important for their function, dendritic cells do not really depend on autophagy to survive. However, very little is known about the role of autophagy for the homeostasis of Langerhans cells, which are antigen-presenting cells of the epidermis. So Langerhans cells, they form a network throughout the epidermis, and their role is to capture antigens and present them in lymph nodes to mount an adaptive immune response. So they are the first sensors of inflammation in the epidermis. And the Langerhans cells, they are seeded from embryonic precursors very early on during development. And they stay in the epidermis throughout the life of the individual because they have quite a long life, and because they, by local proliferation, they divide themselves. And interestingly, Langerhans cells, they have to adapt to a very stressful environment because in the epidermis, they are exposed to radiation and they have low availability of nutrients and oxygen. So we hypothesize that autophagy could be interesting for Langerhans cells to help them adapt to these stressors and live long. So we created a model, the CD207-ATG5 knockout mice, where ATG5, which is a gene that is essential for autophagy, is knocked out in CD207 expressing cells. So these cells are the Langerhans cells in the epidermis. So there is a specific deletion of autophagy in Langerhans cells that we could confirm by immunofluorescence stainings. So you can see in the ATG5 wild type Langerhans cells that there is formation of LC3 positive autophagosomes in the puncta here, and they are no longer formed in the ATG5 knockout Langerhans cells. And when we quantify the autophagic flux, we saw a complete ablation of this flux in the knockout Langerhans cells, which confirms that our model is autophagy deficiency in Langerhans cells. So strikingly, as we started to look at the percentage of Langerhans cells in the epidermis over time, we saw that the Langerhans cells were disappearing from the epidermis, the knockout Langerhans cells. And we could also confirm this by immunofluorescence. So what you see here are ear epidermal sheets, and in white it's the staining of Langerhans cells. And you see that they disappear from the epidermis over time, although not completely. So we started to dig a bit into the metabolism of Langerhans cells and try to see why they were dying. And we observed that the ATG5 knockout Langerhans cells, they had an increased granularity by FACS, and we also saw that they were accumulating lipid droplets. So we could see it by the staining with body P, which is a dye that stains lipid droplets, and we saw that ATG5 knockout Langerhans cells were accumulating them, both by flow cytometry and by immunofluorescence. And then we stained perilipin-2, a protein that covers lipid droplets, and we also saw that the ATG5 knockout Langerhans cells were accumulating perilipin-2. So why could they be accumulating lipid droplets? Well, we also saw that the ATG5 knockout Langerhans cells had increased expression of CD36, which is an importer of free fatty acids. And when we incubated the cell suspension with the stained lipids, so in this case body PC16, we saw that the ATG5 knockout Langerhans cells were also importing more free fatty acids. So they are importing more free fatty acids and they accumulate lipid droplets. We also performed an RNA sequencing analysis, which showed that there were a lot of genes in the knockout Langerhans cells that were upregulated compared to the wild type. These genes were related to metabolism and especially lipid metabolism. So we perform a staining for phospho-AMP kinase levels because AMP kinase is a sensor of the energy levels of the cell. We thought they are accumulating lipid droplets, there is a dysregulation in their metabolism, and we also saw that AMP kinase was more phosphorylated, which means that these cells have low levels of ATP, lower at least than the wild type. And what can the cell do when there is low levels of ATP? Well, this is regulated by phosphorylation of AMP kinase, and the cell can upregulate autophagy to try to degrade components of the cytoplasm and get building blocks to generate energy, which we know our cells cannot do because they are autophagy deficient. They could also increase the uptake of free fatty acids, which we have seen is the case in the Langerhans cells. And another response is also to generate more mitochondria in order to have more beta oxidation and more production of energy. And we stained the mitochondria in the Langerhans cells, and we saw that the ATG5 deficient Langerhans cells had increased numbers of mitochondria. Then we perform a seahorse assay to try to assess the function of these mitochondria because there's more mitochondria, but they still have low energy levels. And we saw that actually there were not a lot of significant differences in the oxygen consumption rate that is allocated to ATP production. So they were not generating more energy. And when we looked at the maximum respiratory capacity of the cell, it was very much increased in the ATG5 knockout Langerhans cells, which makes sense considering that these cells also have more mitochondria. So more mitochondria means more maximal capacity, even though they are not really using it to generate ATP. So we have all of these metabolic impairments, but we still didn't get to why the Langerhans cells are dying. And the fact that they were accumulating all of these lipids made us think of a type of cell death that is called ferroptosis. So ferroptosis is a programmed cell death that occurs when peroxidized lipids accumulate. So polyunsaturated fatty acids in the membranes are very prone to oxidation in a reaction that is catalyzed by iron. And when they accumulate in the membrane, they make it very unstable and they end up opening up pores in the membrane, which is what kills the cell. And what is important about ferroptosis is that it depends on iron. Important mechanisms that are in place in the cell to avoid this toxicity are, for example, the glutathione pathway, which is dysregulated in a lot of models where we have observed ferroptosis. And also very important for ferroptosis is the presence of iron in the cytoplasm. So the transferrin receptor mobilization to the surface is a hallmark of ferroptotic cell death too. What we saw in the ATG5 knockout Langerhans cells was that there were a lot of genes that were related to this ferroptotic pathway that were upregulated. And this included, for example, genes related to glutathione synthesis and the cysteine importer that is necessary for glutathione synthesis, and the iron importer, this transferrin receptor. The mobilization of the transferrin receptor to the membrane was also confirmed by flow cytometry. So the cells had increased expression of this iron importer in the membrane. And in accordance, they also had higher levels of intracellular iron, which could explain why they are dying of ferroptosis. But this is not yet enough to confirm that there is a ferroptosis going on in a cell. We also need to check that indeed there is more lipid peroxidation. So we used a dye called C11-BodyP that measures the production of lipid rows inside of the cells, and we saw that indeed the ATG5 knockout Langerhans cells, they have increased levels of lipid rows production. And when we treated the cells with a known ferroptosis inhibitor, which is called ferrostatin, we saw that this increase of lipid rows production was ablated in the treated cells. So finally, in this paper, we have described a model in which when we knock out ATG5 in Langerhans cells, they are no longer able to perform autophagy. This causes lipid droplets to accumulate. And also, the Langerhans cells are not able to obtain free fatty acids for energy from these lipid droplets because they are not getting degraded. The Langerhans cells have thus less levels of ATP, and they phosphorylate the AMP kinase. And the downstream cascade of the AMP kinase increases the import of free fatty acids, which is exacerbating this lipotoxicity. They are also importing more iron, and together with the accumulated lipid droplets, this is causing the accumulation of oxidated lipids and the death of Langerhans cells by ferroptosis, which explains why there was a depletion of the Langerhans cell network. So with this, I would like to thank you for your attention, and I would like to thank all of the people that participated to this article, especially my supervisors, Dr. Vincent Flacher and Frédéric Gros, and Dr. Flora Narbugas, which was the first author with me. Thank you very much.
Thank you very much for this nice presentation. Maybe I can introduce the second speaker, Ruth Lubin. So Ruth Lubin currently does her PhD at the Dr. Simon Yoner's lab at the Hebrew University, where research is focusing on human dendritic cells subsets. So please, Raquel, the stage is yours. We are looking forward to your presentation, and there will be a Q&A directly for both presentations after your presentation. Thank you. So you're muted, Ruth, please. You have to unmute.
Sorry. Thank you for the introduction and for the opportunity to present our data at today's webinar. It's my honor to share with you a recent paper that was published in JXMed, which explored the lifespan and role of human dendritic cells in health and inflammation. The mononuclear phagocyte systems consist of resident macrophages, circulating monocytes, and dendritic cells, or DCs for short. What distinguishes DCs from the other immune cells is their ability to act as a bridge between the innate sensing of pathogens and the orchestration of adaptive immunity. Under healthy homeostasis, DC comprises two distinct lineages, the plasmacytoid dendritic cells, or PDCs, which are known for their capacity to produce type 1 interferon in response to viral infection, and conventional dendritic cells, or CDCs, which can be further categorized into three groups: CDC1, DC2, and DC3. CDC1 can efficiently prime T cells through cross-presentation. DC2 are known for their ability to induce CD4 positive regulatory T cells and polarize T cells towards Th1, Th2, or Th17 development, depending on environmental cues. Like DC2, DC3 have similar functions, but they're understood to have a distinct ontogeny, and they exhibit several similarities with classical monocytes. Lastly, another member of the DC family is the AXL positive, Siglec6 positive DCs, or ASDCs, also designated as pre-DCs, DC5, or TDCs. These cells are specialized in stimulating naive T cells, and they're speculated to be the precursors of the CDC population. Previously, it was shown that in mice, DCs are a short-lived population. Specifically, one study showed that following the continuous administration of BRDU to mice, splenic DCs showed rapid input of labeled cells without any detectable lag, indicating either immediate generation from dividing precursors in the spleen or rapid replenishment through the bloodstream. Another study established parabiosis between mice expressing the two distinct CD45 allotypes. The exchange of blood-borne cells between parabiotic mice is directly related to their half-lives in the circulation. Cells with long half-life in the blood are equally distributed in the two partners, whereas those with short half-life do not. Lymphocytes in the spleen and lymph node equally distributed in the two partners, reaching equal distribution by day 9 after the initiation of parabiosis. In contrast, DCs never reach 50%, indicating their short half-life in mice. Despite the crucial role that DCs have in our immune system, the lifespan of human DC subsets during healthy homeostasis remains unclear, and translating these data from murine to human is challenging but necessary. Therefore, in order to investigate the lifespan of human CDC subsets during healthy homeostasis, we use the non-toxic stable isotope labeling approach, specifically by using deuterium-labeled glucose. Deuterium is a stable isotope of hydrogen, which can be stably incorporated into the backbone of DNA in dividing cells, and therefore it acts like a birth certificate. Following the administration of deuterium-labeled glucose to healthy volunteers, we analyzed sorted DC subsets at sequential time points, looking for DNA deuterium incorporation by mass spectrometry. And this work was conducted in collaboration with Professor Mikalin's lab. Although it's very difficult to fully appreciate how remarkable this little data point is, it is very striking to see that ASDCs appeared in the circulation as early as 6-hour post-labeling, suggesting a very short post-mitotic dwell time prior to entering the circulation. These ASDCs then followed a similar kinetic trajectory to the other CDC subset, which appeared 1 day post-deuterium pulse. Labeled ASDCs and CDCs continued to accumulate in the bloodstream before starting to disappear from day 3 onwards, likely by migrating into peripheral tissues, although cell death in the circulation might also explain the observed loss of labeled cells. To gain further insight into these kinetic patterns, the deuterium data combined with additional datasets were analyzed using several mathematical models designed to predict the lifespan of each of the CDC subset. This part of the study was performed in collaboration with Professor Becca Asquith and Dr. Jonas Meckerod. After generating few possible models with varying trajectories and performing the appropriate calculation, the most likely model with the highest biological relevance was ultimately chosen. Altogether, these data predict the average circulating lifespan of CDC1 to be at around 1.3 days, ASDCs for around 2.1 days, and DC2 and DC3 for 2.2 and 2.3 days, respectively. These data also suggest that ASDCs are the precursors of the DC2 population, but not of CDC1. So far, we've shown what is happening during healthy homeostasis, but to investigate the role of the circulating DC compartment during inflammation, we used the human experimental skin blister model to sample infiltrating cells. Compared with clinical samples, a noticeable advantage of using human experimental models of inflammation is the ability to track and trace the inflammatory response from its onset through to resolution. To examine the function and kinetics of human DC subsets in response to bacterial insults, dead E. coli were intradermally injected into the forearms of healthy volunteers. This insult provoked a conventional inflammatory response, including heat and redness, which was evident through increased blood flow measured by laser Doppler. Not surprisingly, skin biopsies taken 24 hours after E. coli injection revealed considerable leukocyte infiltration. To assess the composition of this infiltrate, negative pressure blisters were formed over the injection site 24 or 48 hours after E. coli injection, and the blister content was then examined by flow cytometry and single-cell RNA sequencing. It was possible to identify all 5 DC populations in the blister by 24 hours. Curiously, we observed a considerable influx of ASDCs into the inflammatory sites compared with their blood counterparts. Together with the upregulation of the co-stimulatory molecule CD80 on blister ASDCs, these data suggest a unique role for ASDCs as an effector cells in their own right when recruited to the inflammatory site. This is as opposed to the previous perception that ASDCs act only as precursors to the CDC population. To further investigate this notion, we compared the differentially expressed genes between blood and blister ASDCs, and as expected, identified several pro-inflammatory genes that were upregulated in blister ASDCs. In contrast, a dramatic reduction was observed in several HLA molecules. Few of the upregulated genes in blister ASDCs pointed towards an activated interferon beta pathway. Therefore, we tested the impact of interferon beta on ASDC biology, in particular on their antigen presentation capacity, a pathway predicted to be significantly reduced in blister ASDCs. Following the incubation of ASDCs with recombinant interferon beta in vitro, a trend emerged showing reduction in HLA molecules that reached significance with HLA-DQ. This trend was less pronounced in the other CDC subsets. In addition, the presence of interferon beta reduced the capacity of ASDC to stimulate the proliferation of naive allogenic T cells in the mixed leukocyte reaction. Interestingly, these T cells also produced more IL-10. Taking together, these data demonstrate that ASDCs can adapt to become more than precursors and adopt an inflammatory phenotype with potential effector functions. To conclude, we established the transient lifespan of human CDC subset and their plasticity in regulating immunity. Lastly, I would like to acknowledge our lab member and my PI, Dr. Simon Yona, who supervised all this work, and of course, all of the co-author of this paper. I would like to thank our funders and all of our volunteers who made this work possible. I hope you find this insight of our paper, this recap of our paper insightful, and I thank you for your attention and time.
Thank you very much for this nice presentation. So now the two presentations are open for questions that you can actually write into the chat part. Maybe I can start with a first question to Raquel. So Raquel, you make this model where autophagy is deleted in Langerhans cells. Do you know any conditions where autophagy might be impaired in Langerhans cells and then modify immunity?
Well, actually, we know already of a model in which we have a similar phenotype of Langerhans cells. It is a model of imiquimod-induced psoriasis, where the Langerhans cells accumulate lipid droplets. And in these same papers, the authors well, they didn't actually knock out autophagy, but the authors saw that there was a correlation between autophagic genes that were dysregulated in the Langerhans cells of imiquimod patients, and also in the Langerhans cells that were accumulating lipid droplets in the mice. So there might be a link with the pathology of psoriasis, although it's not very clearly established.
Okay, thank you very much. Because I think if autophagy is it's linked to psoriasis, do you think it can really alter the presentation of autoantigen via Langerhans cells and then perturbate the priming of T cells?
Well, we didn't study antigen presentation in Langerhans cells, and to my knowledge, the role of autophagy or autophagic proteins in antigen presentation in Langerhans cells hasn't been studied, but it would indeed be very interesting because sometimes when an antigen is uptaken by an antigen presenting cell, there is a pathway where the autophagic machinery helps present this antigen. And this has been shown in dendritic cells and macrophages, but not yet in Langerhans cells. So it might well be.
I think what would really be super interesting, Raquel, would be to check for granulomatous diseases. There, I would really see a role for the pathway that or for the circle, the vicious circle that you just introduced. Any kind of granulomatous disease in the skin. I also have a question to you, but first maybe to Ruth. Also beautiful presentation, both of you, really super interesting and to the point. My question to you would be, do you find ASDC also in normal tissue without any activation capacity? And then directly to that, I think it's really interesting that they downregulate HLA molecules. Is that an effect that could occur during tissue penetration and eventually pre-maturation? Or is it really linked to the activation with your cool E. coli stimulation approach?
Okay, so without any inflammation in the skin, ASDC should not be there. I think in wound healing models, they are present. And I think that the HLA-DR reduction might tell us something about them trying to regulate the immune response, especially as it's 24 hours after the beginning of the insult. So it could be that they're trying to already wrap things up.
So you think it could be a question of dynamics and kinetics also?
Yes.
Yeah, makes sense. Raquel, may I ask you also? I think it's super cool, a very nice circle that you built up there. Of course, the next question would be about, or actually, I have two questions. One is about, can it be compensated? So if you identify that your Langerhans cells get enough energy, could be by ATP or carbohydrates, so a non-lipid source, could that in a way at least diminish the intake or the uptake of lipids? Do you have a speculation or data on that?
We don't really have data on that. We did see that they were also trying to uptake more glucose because they had increased expression of glucose transporters, but this wasn't enough to compensate the decrease of energy levels. And I think that depending on the environment of the cell or the type of cell that we are talking about, a cell might rely more on glucose metabolism or in fatty acids metabolism to produce energy, and Langerhans cells rely more on fatty acids metabolism.
Yeah. And last question, shortly on generalizability, Raquel. Do you think this is a specific effect that you observe with autophagy in Langerhans cells, or is it generalizable to eventually, at least, let's say, antigen-presenting cells or even beyond?
It was the goal of my thesis to study the role of autophagy in tissue-resident macrophages in general. We just started with Langerhans cells. We know that autophagy is necessary for a subset of peritoneal macrophages for survival. It might be that autophagy is important only in a certain context. So, for example, it's important for the Langerhans cells because they are in this stressful environment, and maybe for other macrophages that rely on beta oxidation and fatty acid metabolism, and that are also in a stressful environment, it could also be important. But in any case, I think macrophages are a very interesting population because they are long-lived. So it's probably more important to adapt rather than disease that do not live that long. So
okay, thank you very much for your presentation and answering so nicely the questions. So now we move slowly to the end of this ESDR kitchen. So maybe have the next slide. So I would like to make a very important announcement. For the next ESDR Kitchen episode will be presented by Sabine Hemming, a former ESDR president. It's my pleasure to announce this talk from her that will be about the misregulated wound healing disease promoting principle that will take place on the 12th of February at 1:30 as usual. And we are very much looking forward to her talk. I would like to also emphasize that all webinars are available online. You have just to go to ESDR Kitchen online. So do not hesitate to watch them again. And I would like to advertise for the next Future Leader Academy organized by the ESDR that will take place in Nice in France. It will be the first time in France, so I'm very happy to announce that. I will be sharing this ESDR Future Leader Academy with Sarah Brown and Knut Schickel. We have already a very nice list of mentors and application to attend this ESDR Future Leader Academy will start on the 3rd of February and application will close on the 17th of March. So I really want to emphasize that all young PhDs and MDs are invited to attend this Future Leader Academy to apply for it. So yeah, this being said, I would like to close still a very important announcement. The 54th annual ESDR meeting that will take place from the 10th to the 13th of September in Antwerp in Belgium. And we are all looking very much to all of you to attend this meeting in Belgium. Thank you very much and see you next time.